Hello, everybody. I'm Bill Kasman. I'm the founding director of GeneWorx Capital and want to welcome you all to another edition of the GeneWorx Discovery Series, where we feature science, people, and ideas from the leading edge of life science. I want to do a special shout out today for those of you joining us from the Boston SynBio Meetup group with yesterday's announcement by Governor Baker of the May 29th reopening of Massachusetts. I'm really excited to meet you this summer and fall at local in-person events. Over the past year, we've all become much more familiar with the science of antibodies and vaccines. Now imagine if you had a synthetic biology platform from which you could select an antibody that could target any disease protein of interest, Alzheimer's, HIV, cancer, COVID-19, the next pandemic, and go one step further. Suppose that antibody had an enzymatic reaction associated with it. Let's call it a catalytic antibody or catabody that dissolved the targeted protein with a single molecule repeatedly attacking and dissolving multiple disease protein molecules. Well, Mother Nature has created these marvelous catabodies over the last 500 million years, and covalent bioscience has created the SynBio platform that enables selection of any such catabody in just months. Today, we have a distinguished panel assembled to discuss the science and potential of catabodies and electrophilic vaccines that can stimulate the formation in the human body. Dr. Anthony Rees, PhD, is Emeritus Professor at the University of Bath, calling in from the UK, and founder of Rees Consulting AB. He has broad and deep knowledge in antibody structure and function. His group was also the first to generate a series of monoclonal antibodies to a constrained peptide antigen and analyze the genetics of the response, the cross-reactivity with native antigen, and the generation of predictive 3D models of the interactions. Dr. Seth Pincus, PhD, is joining us from Montana State University in Bozeman. He's a professor there. His primary research interest is the biological function of antibodies, in particular, the interaction of antibodies with microbes and toxins. He's one of the earliest researchers in vaccine and antibodies to HIV and published heavily in this field. He led reviews of HIV grants and chaired 4 COVID grant review cycles in the past year. Dr. Joel Buxbaum is an MD and professor emeritus at Scripps Research. Throughout his career, he studied the etiology and pathogenesis of age-related human diseases of protein conformation, genetic epidemiology, such as transthyretin amyloidosis using transgenic animals, cell culture, and molecular biology techniques. Dr. Richard Massey is CEO and co-founder of Covalent Bioscience. As COO of iGen, he raised over $100 million in private equity and licensing deals, and he participated in the $2 billion buyout of his company. He is highly knowledgeable in the basic science of catabodies and e-vaccines and has a deep understanding of the unique commercial opportunities for these technologies. And finally, Dr. Sudhir Paul is co-founder and chief science officer at Covalent Bioscience, and he is the discoverer of naturally occurring catabodies. His work as professor at the universities of Nebraska and Texas has brought catabodies and e-vaccines from chemical curiosities to the verge of medical use. He discovered harmful catabodies in autoimmune disease, progressing to devising Covalent's on-demand technology platform for generating catabodies and e-vaccines for disease treatment and prevention. He's authored over 200 original research articles and edited several books. During the first part of our time together, Dr. Paul will lead a presentation with the panelists about the science of catabodies. Following that, we'll have a discussion among the panelists about the challenges, potential, and future of this important technology, and that'll bring us to the end of the hour. However, the panel has agreed to stay after for another 10 to 30 minutes to answer your questions and continue the discussion. Please put your questions as they come up into the Q&A, and I'll collect them and we'll answer them after the discussion and presentation. So Dr. Paul, over to you.
Hi, so without further ado, but with great thanks to Bill Kasman for the introduction and laying the foundation for the discussions of catalytic antibodies, irreversible antibodies. I guess this is addressed to Tony Rees, but others can chime in if they have unique insights. What are antibodies? I felt it is important to establish the molecular structure and their capabilities. So what are antibodies and why are they conceived to be so important in medicine?
Yes, thank you, Sudhir, and hello, everyone. Do we have the first slide?
Yes.
We're all very familiar, as has been said over the past year, more than familiar, more than's good for us, with the whole subject of pathogens, in particular viruses. And of course, vaccines and the development of vaccines, unprecedented speed with which they've been developed. And most of the vaccines induce as a first line of defense antibody molecules. And you see an example of an antibody molecule here in its 3 dimensions. And it has several features. If you look at the picture, those of you who are not familiar with antibodies, what's called the VL, you see the symbols VL and VH. There are 2 copies of that on either side. They hold the business end that binds to, for example, the S protein of COVID-19 and neutralize it. The other end, the bottom end, where you see CH2 and CH3, is involved in what are called effector functions. So when an antibody binds to its target, it can activate via this region certain consequences that can destroy the antigen or remove it from circulation and so on. If we just click forward, Sudhir, one click on you can see that there are a number of different subclasses of this particular IgG. This is an IgG, immunoglobulin G, which is the main production from the vaccine. And just showing that there are slightly different shapes and behavior of these different subclasses, depending on what we need them to do. The interesting thing about antibodies, if you click once more, is that the first line of defense is with what's called the immunoglobulin M. And you see a sort of picture here, which is produced by electron microscopy. And this is a much more complicated molecule, but it turns out, as I said here, it's the first defender. It's called the naive immune response. But it's also the front runner, one of the front runners in catabody behavior. So we'll come a little later to how these can be used and activated for the sort of applications that we're going to be talking about today.
Great. So, Tony, if you could also address a little bit about the complicated history of catalytic antibodies going back several decades, and then in a sort of a presaging of what we are going to discuss, in your own opinion, are they really coming of age after all these decades?
That's an interesting question. And of course, I guess most people who are listening will be familiar with the extraordinary interest that developed in the 70s and 80s in catalytic antibodies made against small molecules. And those were based on what are transition states or stable transition state analogs for particular enzyme reactions, something suggested by Bill Jenks back in 1960.
I think Tony's video is frozen.
Even 20 years before Jenks. The question of naturally occurring catalytic antibodies, of course, is much more interesting for reasons that you will hear about later. And as Sudhir indicated, that was at the end of the 80s that the first example of that. And I was happy to be involved with Sudhir in characterizing this particular light chain catalytic antibody. So I think the coming of age it always takes time. If you remember the 1975 monoclonal antibodies, and the sort of cognoscenti of the time thought it was mildly interesting. Well, it took probably something like 20 years before it became clinically relevant. So there is a sort of incubation period, and I think now is the time where we can see, or will see, the application of antibodies that can be induced based on the natural antibody concept.
Great. So I will try to address that question. Are they coming of age, and why are catalytic antibodies of interest in modern medicine? A conventional antibody is a one-to-one binder. A catabody, by definition, is a catalyst. It turns over, as the familiar ad says, like it's a never-ending energizer bunny. I shouldn't say never-ending. It is finite, but it's many thousands of cycles, so it's multi-cyclic. The antibody, that the business end of the molecule that Tony showed, it is merely a binder, and to remove the target, so target-specific removal is the objective of both ordinary antibodies as well as the catalytic antibodies, the catabodies. An ordinary antibody requires a macrophage, a phagocytic cell, to take up the immune complex, and that's the only way it can remove the target. Whereas a catalyst can break up, as is shown in this picture. It breaks up the target directly, and it goes on over thousands of cycles. If the target is present in high abundance, particularly if the target is present in high abundance, like the amyloids that we are interested in, that's one class of targets for which catabodies have been validated, and there are publications to that effect. Unfortunately, for high abundance targets, inflammation is an ever-present danger. And in particular, in Alzheimer disease, there was much excitement about ordinary antibodies removing the amyloid beta that is thought to be the cause, in quotes, cause of the problem. But it didn't work out because of inflammation. These catalysts do not cause inflammation, simply because they don't recruit the macrophages, they just directly break the target. Now, our innate catabodies, as we call them, they are products of millions of years of evolution. They are legitimately called innate antibodies because they are present in healthy humans. Our job is to exploit, in the company, Covalent Bioscience, and as a professor until recently in my academic work, our job was to exploit these innate occurrence of the catabodies using these electrophilic targets, and we select the best targets. That's a selection from libraries. We can also induce them by immunization, like vaccines. We call them e-vaccines. And we are hoping that with the data that we have in hand, that eventually this will lead to much better therapeutics vaccines, but also diagnostics, in particular irreversible, a different class of antibodies that I will mention, that we will discuss it towards the end of this panel, that they can be used even for diagnosis beyond what is capable for ordinary antibodies. The next issue is, how do we isolate them? So as I say, we use these electrophilic analogs to pick out the specific antibody from a big library. The scaffold, this IgM scaffold that Tony mentioned, it is the most naive, the most innate of the antibody classes. Antibodies are generally known for binding in the IgG class, which is shown here, which unfortunately is less catalytic, but fortunately, it expresses the highly evolved binding function for which these mammalian IgGs have evolved to bind. But our IgGs, they express the binding function in the irreversible form, which can also be thought of non-dissociable complexes are formed with the target. And we think of them as infinite, quasi-infinite affinity antibodies, these irreversible IgGs and the catalytic IgMs. In the IgA format, the constant domains are different for the antibody. As Tony mentioned, the constant domains influence their structure and function, the function also. And the catalysis is seen even in IgA compartment. And these are the variable domain fragments that we published are highly catalytic. The good news with catalytic antibodies, just like regular antibodies, the reason they're such good drugs, the regular antibodies, is they have predictable pharmacokinetics, they stay around in the blood. Our catabodies behave in a similar fashion, they are highly predictable kinetics. We are targeting the brain in Alzheimer's disease. And this panel just shows data that a significant amount of the intravenously injected catabodies, they do enter the brain. So we are getting delivery, drug delivery is happening into the brain. And then we are also interested in systemic amyloid disease using IgMs, these large molecules here. And we are seeing that the target tissue, in this case, which is joints and the heart, that's where the amyloid disease is happening. And that's where we want our antibody to reach. Indeed, there is no hindrance in permeation of the IgM into these target tissues. How do we make these e-targets? We attach what we call an electrophilic target, which is an analog of the natural electrophile, which the catabody and the irreversible antibody recognizes. We simply attach it to the protein target. Non-covalent binding occurs to the catabody. That is the source of specificity. And this electrophile, this non-hydrolyzable, this is not broken. The catabody doesn't, it binds covalently to the synthetic electrophile, but for the natural electrophile, the intention is that the peptide bond, which is shown here, would be cleaved. So this is hydrolyzable, the natural peptide bond, whereas our surrogate, the synthetic electrophile, simply binds covalently, and with this, in this mechanism, we can fish out the catalyst from large libraries. This is a catabody with the 3 amino acids that are responsible for catalysis. It's a serine protease class of catalysis, much like natural proteases. Antibodies have the same catalytic sites, and these 3 colors, in fact, Tony constructed this model way back in the 90s for our first catalytic antibody that we isolated, a monoclonal catalytic antibody. They attack this carbon by forming a covalent bond, and then if a water molecule is present, that hydrolyzes the peptide bond. Okay, so I think the next issue I'd like to very briefly touch on is what are they doing? They're present in healthy people, and we discovered them as autoimmune patients. But our interest now is on beneficial catalytic antibodies found in healthy people. And here is a disease model that we have made that are operating, is the operating sort of hypothesis based on which we design further studies, that in young people, there are catalytic antibodies. The amyloid, which is the target, which accumulates, and Joel will talk about amyloids in a couple of minutes, but the amyloid is minimal. Amyloid accumulates with age. The amount of catalytic antibodies found in humans is insufficient, and therefore we get the disease. And I have 3 remarks, I guess, on the related to this disease model. One remark is that for some targets, in this case, transthyretin amyloid or misfolded transthyretin, as we call it, mis-TTR, essentially all of the hydrolytic, all of the removal capacity of blood in healthy humans is due to these catabodies. The normal enzymes are not very good at removing the TTR. So we think, therefore, that this is the bulk of the proteostasis mechanism, the homeostatic mechanism that keeps the amyloid in control. The amyloid accumulates in the test tube within minutes to hours. It is good news that there are catabodies present in our working disease model, that we stay alive without amyloid disease for decades before the amyloid accumulates to a large enough level to cause disease. So, Joel, I'll hand over to you for your comments on what is amyloid disease and why are amyloids worthy targets for modern medicine.
While I think the history of antibodies is described by Professor Rees, we have to look at historically the same thing is absolutely imperative with respect to amyloid. Amyloid was first described in humans in the mid-19th century, and it was described in pathologic material, and it remained a mystery from that period in the mid-19th century to the mid-20th century until techniques were evolved to solubilize and chemically analyze these collections that were compromising organ function in humans. And after those advances, it was possible to discern to the current date where there are at least 36 different human proteins that are associated with either local or systemic forms of amyloid. But all these collections, or the components of all these collections, have the same shared characteristics. That is, they are made as soluble proteins and then become insoluble during life course under physiologic conditions. When you look at them under the electron microscope, they appear to be as fibrils. It's very difficult to solubilize them. The fibrils have certain characteristics. That is, they bind the dyes Congo red and thioflavin T, and on X-ray diffraction, they all have a similar beta sheet structure. And over the last 40 years, 36 different proteins have been identified with these characteristics that form, that are responsible for discrete human diseases. So I think that, and some of these are associated with more than one disease phenotype. And you can see from this slide that the age, and again, all amyloids, or most amyloids in humans, appear with age. There are none that I know of that occur systematically before the age of 50. And the notion is that after this age, the body's capacities to deal with these potentially insoluble proteins dissipates. Consequently, they can become, either because of mutation or various other modifications, they become insoluble under physiologic conditions, deposit in various organs, and compromise the function of those organs. Now, in the world of human disease and medicine, there are two major forms of amyloid that have presented therapeutic challenges to the biologic and medical community. One is Alzheimer's disease, which is notably the poster child of amyloid diseases. And again, as we all know, while there are early onset genetic forms, most Alzheimer's disease occurs in people over 60 and 70 years old, with current estimates being somewhere that 25 or 30% of people above 80 have dementia related to amyloid deposition. It is not entirely clear, despite 25 years of work, to show that the protein A beta is critical. Amyloid formed from A beta is important in amyloid formation in the brain, although it may not be the etiology. That is, there may be other conditions that cause the overproduction of the amyloid precursor in the brain, and the body has no other way of dealing with it except to make fibrils. It's been a difficult therapeutic challenge, essentially because we don't know when it starts. And it's very clear, at least at the moment, that once the process of amyloid formation starts, it's very difficult to interrupt it because the protein itself creates or incites certain responses in tissues, which initially may be beneficial, but in the long run may actually cause additional tissue damage.
Joel,
So
can I break in, Joel, by commenting partly and also opening it up for a little bit of discussion? The problem of Alzheimer's disease is really a key problem, a national, international, existential problem for humans. And the A beta hypothesis, this is my comment, it has never been tested properly in the clinic, in the sense that all of the antibodies that have been tested, the ordinary antibodies, they cause inflammation. And if you remove amyloid, but you also cause inflammation that is neurotoxic, that there is toxicity due to inflammation, the brain is damaged. So why are we so, what is the basis for the lack of enthusiasm simply because the regular antibodies have failed, is my comment. I think it's dubious. The lack of enthusiasm is dubious from my point of view. I leave my comment at that point.
Well, I certainly think that the lack of enthusiasm at the moment is the result of 4 or 5 large-scale clinical trials having employed antibodies that were shown to be effective in mouse models of Alzheimer's disease. And I think it makes it very clear that what works in the mouse, even in what appears to be a decent model of disease, does not exactly translate into a human benefit. There are many examples of this.
Yeah,
but to be correct to your point, in fact, it's only been tested with conventional antibodies, essentially because they did work in the mouse models, and the notion was at the time that if it worked in the mouse models, which were based on excessive production.
Sorry to interrupt you, but I believe that's a wrong notion because the mouse brain does not get inflamed in the same way as the human. Leaving aside the technical issues, my contention, which is open for obviously debate not only here but in the literature, is that the amyloid hypothesis has not been tested, number one, in an appropriate way. And I think Catabodies offer the first ability to test the hypothesis because they don't cause inflammation, as we will see in some of our supporting data. Second, the downstream effects have not been countenanced. The tau buildup, yes, we can do it separately. We can do separate immunotherapy against tau, but the combination of the two might be the cause of the disease. And certainly tau is important downstream. I'm going to leave the point here. We can come back to it when we discuss whether Alzheimer's disease is the right target for Catabodies for commercialization. Is it a good choice? This is a central sort of point for our company and for our academic thinking. Joel, did you have any final sentence on this point before we move on?
Well, I think your point is well made that these antibodies, catabolic antibodies, have not been tested in A beta. And I think that remains an open question.
Great. With that, then I will deliver to you for your discussion a more, I guess, more scientifically pleasant state, a scientifically amenable disease, which I think is certainly true for transthyretin amyloid disease. And please tell us your opinion on transthyretin amyloid disease in general, and also whether catabodies are appropriate for testing out in this disease.
Well, historically, transthyretin amyloid was originally identified in a small number of families as an autosomal dominant disease, where there were mutations in the normal protein transthyretin that made it unfold and dissociate to produce amyloid precursors. It became clear after a while that there was also an age-associated form of transthyretin deposition, predominantly but not exclusively in the heart, in which the deposits in the heart consisted of fibrils that were made from wild-type transthyretin, that is, transthyretin that had no mutations in it. The basis of this instability is not entirely clear, although several hypotheses have been offered, not the least of which is that aging-associated modifications of the transthyretin protein make it less stable. What happens then is the transthyretin molecule, which normally exists and functions as a tetramer, that is a combination of 4 identical subunits, which carries a thyroid hormone. This becomes unstable and releases its monomeric components, and the monomeric components have the tendency to aggregate, then form fibrils and deposit in tissues. Clearly, what has happened over the last 10 or 15 years is that a number of therapeutic approaches have been tried, some of which directed directly at inhibiting the synthesis of the transthyretin precursor. And essentially, since a normal adult human seemed to be able to function very well without transthyretin, this reduces the amount of protein that's available to misfold. And there are now 2 oligonucleotide therapies available that work on the disease in this fashion. Another approach has been to just stabilize the tetramer with a small molecule so that it no longer is unstable, doesn't release the monomer, so it deprives the body of a misfolding substrate. And by reducing that, presumably, in both of those instances, being deprived of substrate, the deposits in the tissues, which compromise organ functions, will dissolve either pure thermodynamically, that is the transition from soluble to oligomer to fibril will become reversed in the absence of a substrate driving the interaction, or in fact, because there are mechanisms within the body that mobilize the fibrils, generally thought to be by macrophage digestion of the deposits. But this has been to date a failed approach, while the supply side approaches seem to have worked reasonably well, and several of them are now FDA approved. Even these approaches leave residual organ dysfunction.
Thank you. Thank you very much for that. If you could make a final comment, and then I want to switch quickly, in the interest of time, to the catabody aspect. So this is a great.
I think that it is the time for the catabody approach, in that conventional antibodies, again, have pretty much failed to resolve the tissue deposits once they are there without regain of normal function.
So I'm going to then show the supporting data. Thank you, Joel, for the great introduction and your expertise in the field of amyloid disease in general. These are valuable comments that you've made. On Catabodies, I will say, I will not go over the details of the supporting evidence. Much of the evidence is published, but the Catabodies are unique that they can remove the preformed amyloid form. The competing drugs that Joel mentioned, all of them act at the prior steps of amyloid formation, namely the aggregation step, and there are changes in equilibrium as he described beautifully. But the Catabodies not only interfere because they break up the target in a very highly specific way. The transthyretin target is exquisitely broken by these naturally occurring catalytic antibodies. Essentially, as is shown here, they make the amyloid disappear. The particles become soluble, both using synthetic amyloid, which we tested in the test tube, and then the tissue of a human, with a patient with spinal stenosis who had amyloid deposition in the vertebral tissue. The brown color after treatment ex vivo with the Catabody essentially disappeared completely. And this is a completely unique approach. Joel, we talked about Alzheimer. Joel mentioned the need for a new approach. The classical approach with antibodies and small molecule drugs is not working. I'm just going to make declarative statements again for published data about Catabodies in order to provide the context for our enthusiasm to develop these Catabodies commercially, or to deliver them to the clinic and test them out further. So we observed that we are able to remove the amyloid seen in the brain. These are brain tissue sections. The cortical A beta is reduced by 40% or so. We also reduced the soluble A beta species by at least 30%. The remarkable feature is that this is a 10-day treatment, unlike conventional antibodies that require phagocytes to remove the amyloid beta. Months of treatment is necessary. And in this particular model, this is the model that best simulates human inflammation in the literature. This unique model, the Catabodies are working. The drugs, like ordinary antibodies, the misleading data with ordinary antibodies came from models that generally do not show much inflammation, and they're accepted as rather poor models of the inflammatory disease that is found in humans. We saw no inflammation seen by microglia, which are the phagocytes, and similarly, we did not see the effect of the inflammation, which is vascular leakages that show up in patients treated with regular antibodies. We did not see it in this inflammatory mouse model. The rate of Catabodies since Linus Pauling, I'm sorry, not 70, since Jenks, and then also since my own discovery of Catabodies, the naturally occurring Catabodies since 1989, the rate of the catalytic turnover is a key. And these innate Catabodies that we isolate from large libraries, the best ones that we want to use for therapy as supplementation for the natural defense function of the Catabodies in blood, they have impressive rates, far more than is required. So this is the rate, turnover number, and that is sufficient to predict. As we saw in the ex vivo study, we saw complete clearance. We can remove with nanomolar amounts, nanomolar concentrations of the Catabody, large amounts of the target amyloid. And with that, I think we are ready to sort of switch gears. And the gear switching for the panel and for the audience, I'll remark that this gear switching is not by design. In other words, this is how the discoveries were made of the importance of catalytic antibodies. We had funding from NIH for making a vaccine and making catalytic antibodies to HIV. I'm going to hand over to Richard to describe briefly our e-vaccine, but more important also to lay the foundation to simply give us his evaluation of why don't we have a vaccine to HIV despite 40 years of intensive devotion of intellect and money. Billions of dollars have been spent by NIH and the rest of the world in making a vaccine, and we don't have one. We think we have it. We have a promising candidate. Richard, please take over.
Yeah, thanks, Sudhir. Well, I think that the reason we don't have a vaccine today for HIV is that the virus itself poses an unusual and unique challenge in terms of developing vaccines. As we know, the HIV virus mutates at a very high rate, and it integrates into our DNA. When we get infected, if a person getting infected with HIV is essentially infected for life. Many viral infections, our immune system clears the infection, whereas in HIV, we don't clear the infection. And I think a key factor in what happens here in the infection process and why vaccines up until now have failed is the immune response doesn't really target what we call the Achilles heel, an essential part of the virus that's essential for binding to a receptor on a cell and causing the infection. And if you look at the right-hand panel, I think that we can basically see that, you know, a lot of the vaccines have been tried and have been many decades of work going into clinical trials. Those vaccines haven't actually induced an antibody directed to the Achilles heel. And one of the reasons is we don't see class switching in terms of moving from an IgM response to an IgG response. And you know, that's what's kind of fueled the research over many years. Basically, it's a variation on a theme. Many companies and the National Institutes of Health have continued to develop vaccines using the same principles over and over again, have been never able to induce an antibody that's directed to the Achilles heel. And what we discovered at Covalent was that by chemical modification of a protein in the HIV virus, we could actually target the Achilles heel and cause that class switching to occur. And in animal studies that we've done, we've shown that we can induce very broadly neutralizing antibodies using our HIV vaccine, something that's never been achieved with other vaccines to date. And I think Seth can probably do more comments on the science behind this.
Yeah, so I think one of the key points that needs to be emphasized about this whole e-vaccine approach is that with most vaccine approaches, you throw in the antigen. In this case, it would be the HIV envelope protein, and the immune response develops accordingly. Here, we can specifically target a key epitope that allows for focusing the immune response on this key structure. And I think that was what's seen in the IgM, IgG loop, and then the ability to get broad neutralization because that is a key structure. And so I think that's one of the big advantages of this approach and what makes it exciting.
Let me, thank you, Seth. I'll supplement that only by mentioning that HIV has found many ways to evade the human defensive immune response. One of the ways Richard and Seth mentioned, which is class switching to that particular epitope does occur. Here's another way. We got, when we immunized with the e-vaccine, this is animal model data, we got very nice catalytic IgMs, so very robust catalysis. but they never neutralize the virus. So the contact with the epitope, despite catalysis, is no good because contact with the epitope doesn't occur. On the other hand, we induced irreversible IgGs, which is these here. So the catalysis is much lower with these IgGs, as I mentioned at the outset, irreversible antibody, irreversible binding to HIV, to this epitope is much better. And this is the one that the irreversible binding then is the vaccine principle, not catalysis. Having said that, and I'll open up the floor to for COVID, which is the next subject we wanted to touch on very briefly, said IgM and IgA are perfectly valid vaccine-induced antibodies, which are, and these are the two classes of antibodies that can display catalytic activity robustly. So although with HIV, catalysis is useless because of the contact problem, from the literature, it appears to me that it should be feasible to develop a Catabody vaccine against COVID for better control of the pandemic, including the new variants. And if you could comment on that point, Seth, and then we'll close this phase of the discussion and move on to the more general discussion. And yours is the final comment on COVID.
On COVID, huh? So we're watching the evolution of an epidemic in real time, and anyone who says they know what's going to happen is full of it. Basically, the variants that have arisen in COVID at this stage of the game are primarily variants that have arisen as a result of the variant viruses replicating faster or being more infectious. We haven't yet seen what happens when the immune system is the selecting agent. But now that we have large populations of people who are immune to the current versions of the virus, we'll see what immune selection does and whether we're dealing with a similar problem as with HIV. The one thing I would just add to what Sudhir said vis-a-vis HIV is, yes, we've spent 40 years and billions of dollars to make an HIV vaccine, but as it turned out, all of that work went into being able to make a COVID vaccine in 6 to 9 months, record time. So we were aiming for one target, but all of the stuff we learned has been applied to a new target, and there's no reason to believe that the catalytic and irreversible antibodies would be any different. And so obviously, I view that there's promise for them in this field.
Thank you. With that uplifting sort of statement, a positive statement, I hand over to you for the remaining time, Bill, and for the general.
Maybe to hear before we move to the general questions, maybe a comment on the more recent developments by the CDC to basically, say that people that are vaccinated are safe, that they can't be reinfected. I think it would be worth a comment on what's going on here with the variants that are rising around the world, and the statement about being vaccinated makes you safe.
Right. So let me venture the comment, Richard, and thanks for stating it the way you did. I think the limitation, antibodies have fundamental limitations which are imposed by their binding affinity. If the binding affinity for the variant is not large enough because the mutation has occurred, the variant will be poorly neutralized or not neutralized at all, as has been documented recently, both for the South African variant and the Indian variant, which is 6-fold less than the original Wuhan variant, 6-fold less the Indian variant, and which has reached the shores of the United States already. That's the intrinsic limitation. Essentially, catalysis and irreversible binding, they offer ways to overcome that limitation, because very quickly, if catalysis occurs, then the binding affinity doesn't have to, the immune complex does not have to be stable to maintain the neutralized state of the target virus So I think it's a reasonable thing to say that catalysis and irreversible antibodies may be particularly suited for targeting the variants.
Could I just have a quick comment, Sudhir, on that? I think that's a valid point. And I think the thing to remember is that for an antibody, a normal antibody, reversible antibody, to do the blocking with a potential escape mutant, it has to block the binding to its receptor, the ACE receptor. With a Catabody, if you target another region of the protein that, if you like, destroys the protein, then that the virus has to generate mutants in an area that it may not want to do, or for various reasons. So I think you have a much larger flexibility when you're trying to disrupt a protein chain. You don't have to go for an exact position that is at the interface of an antibody and its receptor.
Thank you. Bill, over to you.
Sure, just kind of building on that point with mutable viruses and the opportunities for using electrophilic vaccines to induce catabodies in humans. I want to ask Seth again, what do you see as potential major challenges or opportunities for e-vaccines, for which e-vaccines might be a uniquely powerful solution versus other techniques that are being used today? And we spoke about COVID and HIV, and are e-vaccines a unique approach to that would be more powerful? And then what other advantages does it have with other mutable viruses?
Well, as I said, I think the advantage is the ability to target a specific epitope on the structure. And as Tony said, it doesn't necessarily with the catalytic antibody have to be a structure, right in the site where the virus recognizes the cell. And it can be elsewhere and catalytically destroy the structure of the receptor. And you can target areas that have to be conserved. Clearly, our vaccine studies and the futures of vaccines, there are viruses that seem quite amenable to making vaccines, as we've seen with COVID. And then there are viruses that have proven quite resistant. Obviously, HIV is the example there, but there are a number of other diseases that can result and we don't have effective vaccines for. And I think that's where the catalytic antibodies or the vaccine approach really has the most potential, is in the viruses that have resisted the development of effective vaccines.
What would your top 3 shortlist be to name a few of those? Is that putting you on the spot?
Well, certainly dengue worldwide would be a target. We've had difficulties developing a dengue vaccine because of the possibility of what's called antibody-dependent enhancement of infection has scared the bejesus out of a number of vaccine makers. We have a couple of vaccines that are being tested and appear to have some efficacy, but dengue would be high on the list. Clearly, an emerging coronavirus that would be different than COVID too. There's a lot of concern. Are we going to be ready for the next coronavirus epidemic? And so if we can pull out structures that are common amongst all of the coronaviruses, we can be prepared so that when a new one comes along, we already have the vaccine.
Seth, can I chime in and vote for Staphylococcus aureus, or not a bacterium? Simply
Well, I thought we were sticking with viruses.
Sorry, I was just going to complete a quick comment. Simply because we have some preliminary data, it's early days. We have not tested out the vaccine approach for many things. So So for Staph aureus, the superbug, there's a glimmer of hope there, and that's why I just wanted to vote for that on your list.
Okay, well, if we weren't going to limit it to viruses, then I'd certainly put TB and malaria way high on my list for trying to develop an effective vaccine. They affect huge populations around the world.
Okay, and we're running up near to the end here. So I want to do a lightning round, go around to everybody, and give me your top challenge and best opportunity for Catabodies and e-vaccines in your estimation based on your background. And this is a lightning round, so short answer, and we'll test Dr. Paul on that. So you're first, put you on the spot. Top challenge and top opportunity.
I'm going to be driven by my bias for preliminary data. I just showed a slide. If you can see it, I vote for transthyretin simply because it is a relatively simple target for which drugs have already been validated.
Okay, TTR it is. I'm going to keep it. It's got to be a lightning round.
Thank you.
Okay, Joel, what's your thoughts?
Well, I have to go with TTR as well. I think because the Catabody approach is very good at targeting tissue dysfunction. And I think that's where our major gaps are now. Even with suppression of the supply side, tissue dysfunction remains.
Okay, let's move over to our infectious disease and antibody folks. Dr. Reese, where do you land?
Yeah, I've just been writing finished the chapter on coronaviruses, although of course the story hasn't finished in my new book on the history of vaccines. So I'm going to vote for coronaviruses. There's SARS, there's MERS, which is a 30% mortality rate, and there's of course SARS-CoV-2, and targeting a conserved region with a Catabody.
Excellent. And Dr. Pincus?
Top challenge, I'll go with HIV because no one's cracked it yet. Best opportunity, amyloid is such an important process in disease pathogenesis. The ability to deal with it in whatever form would be a huge advance.
All right, excellent. And we'll to wrap up with you, Richard, and I know there might have been a slide you wanted to share. We have 2 minutes, and then just before Richard does his final comment here, wanted to let everyone know, put any questions you have, put them into the Q&A of Zoom. I've been collecting them as we go, and we'll go into a Q&A session after Richard's comments.
So just to answer the challenge question, I think, we face a general challenge because we have a new technology, catalytic antibodies, irreversible antibodies, and e-vaccines. And the challenge is really to demonstrate efficacy in a clinical model, not in the animal model, but in actual patients. And I think our closest, the ability to get to the clinic quickly and demonstrate efficacy, I think is going to be with the catalytic antibody to TTR for treating amyloid disease. I think there's some orphan indications there which we can target and move into the clinic very quickly and demonstrate efficacy with the antibody. And demonstrating efficacy with an orphan disease gives us the opportunity to broaden the indications to other diseases associated with TTR amyloid. And then just focusing on this particular slide, I think that having a platform technology provides a lot of challenges because you really need to focus on the areas where you can move quickly to the clinic, convince investors to back the company to support clinical trials. And with the technology that we have so many different opportunities, whether it's vaccines for HIV or COVID or Staph aureus, or the catalytic antibodies for treating other types of diseases. And we even have an opportunity for using antibodies, the irreversible antibodies for diagnostic purposes, which could move quickly into commercialization. So I think this slide does a nice job at summarizing the various opportunities, but the company needs to really focus on developing use of an antibody where we can demonstrate efficacy very quickly, and I think that's the TTR antibody.
All right, excellent. Well, I want to thank all the panelists and thank the audience for attending. We're going to move into a Q&A session now. And again, a lot of world-class talent and expertise here. So really appreciate the comments and look forward to continuing the discussion here just a little bit further. So we'll take questions from 10 to 20 minutes. And we'll start with this first one. Is it feasible to develop Catabody cocktail to neutralize the severe COVID infections? Who would like to take that?
Yes, it's feasible. A cocktail would always be better than an individual antibody. It's been tested multiple times with COVID antibody cocktails versus others. They would have to design and pick the target sites appropriately, but certainly feasible.
And I know we said it, I think it might be worth just summarizing in 1 or 2 sentences one more time. I'll throw this to you, Richard. If you could describe the difference in the way a Catabody would attack the COVID-19 virus versus an antibody approach.
Yeah, I think that, I mean, has been highlighted by Seth, and is the fact that the conventional vaccines are targeting a region of the spike protein on the coronavirus that's going to block basically binding to the receptor. And we know that the virus is mutating, and that, as Sudhir mentioned, some of these antibodies don't bind with high affinity anymore. And one of the concerns is that the mutations could actually result in antibodies not binding at all to the receptor binding domain. So the unique advantage of a catalytic antibody and an irreversible antibody is that a catalytic antibody could actually bind the spike protein in a different region. It doesn't have to bind necessarily the domain that's involved in receptor binding, and could possibly inactivate the virus by just simply enzymatically breaking up the spike protein. So we think that's a unique advantage that catalytic antibodies have, and could really play a very important role in preventing infection by variants. The irreversible antibodies offer another feature with the high binding affinity. They essentially bind with infinite affinity. And we think that's a particular advantage in neutralizing the variants. So it's these two features. And a vaccine would induce both those functions. They would induce irreversible antibodies and catalytic antibodies, and those dual functions could really make it a very powerful vaccine to protect us against the variants.
Excellent. And then there was another comment from that questioner, which is the COVID cocktail is the need of the hour for sick ICU patients. So I guess that puts the pressure on the team. How do we get that done? Let's circle back to Joel. I know you picked TTR as sort of your top opportunity. In your opinion, are there targets for which Catabodies are a bad choice, and how would you characterize why?
There is one possibility that we think about, but I think the Catabodies may actually get around it. That I mentioned before that, at least in the case of TTR, if in fact reducing substrate allows the deposits to solubilize thermodynamically, it is possible that the molecules released, the fibrils, may be the least toxic form, and that the dissolution may yield the release of molecules that re-oligomerize and may actually be toxic in a different way. I mean, most of the amyloids, or at least the ones that have been studied adequately, that the precursors seem to have two modes of toxicity. One is early from oligomers that have direct cell toxicity, and that's certainly been demonstrated in tissue culture. But the longer term effects are the compromise of organ function by fibrillar deposits, who may no longer be toxic in their own way, but take on a different aspect of organ compromise. So the question is, if you solubilize at least the fibrils, will they in fact release molecules that are toxic? And that is, there are no data that address that question in a direct way.
Okay, thank you. Here's another question. I'll maybe ask Tony to respond to. Given that all that we've heard about IgM and IgA being good catabodies, is a therapy-grade rapid specific IgG catabody possible to isolate from nature, engineer, or induce by e-vaccine technology?
I think that's a good question. There is a big interest, of course, as you heard from Sudhir, immunoglobulin A, which of course are located at the front end of viral attack at mucosal surfaces. There's a big interest in vaccines that target the mucosa, and with IgA as a really great candidate for catabody activity. I would, and the interest in developing ways of delivering vaccines, in particular adjuvant compositions that activate IgA cells rather than IgG. So whether you would go the IgG engineering route, there is some evidence that the particular constant regions of IgA and IgM have an influence, a strong influence on the catalytic activity, which is possibly why you see lower activity with IgG than you do for IgM and IgA. So I think that's something to be worked out, but I think there is an interest anyway in inducing vaccines via mucosal challenge. And I think that might be an interesting way to go, particularly for viruses.
And Sudhir, as you add your comment onto that, I see you're going to, could you also speak to manufacturing issues with catabodies? Because I think it's somewhat a related topic.
Exactly. So my remark first on the company and IgG versus IgM is that manufacturing facilities for IgMs would have to be rigged up. It's not very difficult. The procedures are available, but the large-scale facilities for IgG manufacturing, big industry, 95 billion per year approximately. Most pharma companies would prefer to use their existing facilities, and it's a substantial cost to manufacture the IgMs or the IgAs. So that's a general issue. On the issue of IgG catalysts themselves, can we isolate them? In nature, if depending on the target, we have evidence that IgG catalysts are rare, but when they are present, those rare catalysts, they can express high activity as IgGs. So we've just cottoned on to the low-hanging fruit for monoclonal therapy. We take the easy ones to isolate, the IgMs and the IgAs as the catalyst. If we go back to the drawing board and use high throughput mechanisms for library selection, I think high turnover IgGs, rare ones, we would catch them, and we are actually close to doing that for one target, which is the reason for the confidence that I'm expressing. They're rare, but if you use flow cytometry to isolate the rare antibodies, then you get IgG high turnover catalysts. So it's just more difficult to get catalysis as IgG, but it is not impossible. That's my overall comment. In addition to the engineering that Tony mentioned, that engineering can be done. We have, in fact, 2 publications on that one could engineer IgGs with some catalytic activity, with some level of detectable activity.
Okay, and then I have a final question, I think, for Seth, and anybody else who has knowledge to chime in. There's a lot of familiarity with mRNA vaccines with Moderna and Pfizer and the COVID vaccine. How would you compare that technology to e-vaccines and pros and cons in terms of what it can be applied to or efficacy or any other way you might like to contrast it?
Well, I think
Development time.
It's related to the essential difference with the approach. The mRNA vaccine, you're targeting whatever that mRNA encodes, whereas with the e-vaccine, you're focusing the antibody response on a specific target that you have chosen. And so in that regard, I think is one of the key differences. And of course, the types of antibodies that you elicit out of those responses will be quite different as well. And so I think the mRNA is a good platform for the viruses that it works for. But I mean, they've obviously tried it with HIV. That's where it was developed for, and it hasn't exactly led us to an effective HIV vaccine yet. And so I think there are just times one will work and the other will work. There may be some overlap.
Well, Seth, isn't the concept behind the mRNA vaccines essentially you generate a conventional antibody response, except that the antigen is synthesized within the organism rather than being injected into the organism? So
Absolutely.
that qualitatively, the immune response shouldn't be any different than conventional, but quite different from the electrophilic process.
Correct. Absolutely.
Except for incidental reasons. There are reasons why the mRNA, so there are rational causes for why the mRNA vaccines induce a higher titer. That's the COVID vaccine experience. The titer is somewhat higher, which is the reason why the Pfizer and Moderna vaccines are superior. The epitope specificity is not going to change, except for incidental reasons where the folding of the protein in situ, meaning in the cell, as opposed to in the test tube, and then taking the test tube vaccine, the protein vaccine, and putting it in there. In principle, there should not be a difference, but the titer is higher for mRNA vaccines. For electrophilic vaccines, eventually one could develop incorporation of electrophilic amino acids into the protein itself by the cell. And Peter Schultz, in fact, at Scripps has published a couple of papers on such transcription systems, as you would say. The technology is not ready for vaccination, but as a long-term objective, one can conceive that if electrophilic vaccines make the commercial grade, someone will find a way to make them into mRNA vaccines.
Okay, and then we got one more question. When and how does the spike protein stop formation in the mRNA vaccine? Anybody? Is that?
Well, I mean, that's actually a good question. If I understood it correctly, and I was just typing an answer, but.
Oh, okay.
Yeah, the mRNA is taken up into the cell. It's a synthetic RNA, and it has a certain amount of inside the cell lifetime, and it's likely that we continue to make the RNA or the protein encoded by that RNA for at least a couple of weeks, but if that's the question, then the protein will persist and continue immunizing for at least that period of time. Vis-a-vis the mRNA vaccines, I think what we have to, when we're comparing this platform versus a conventional protein platform or anything else, is this was the first time mRNA vaccines have been used. And I think the companies did not want to under immunize. I think they erred on the side of a much stronger immunization than may have been necessary, and that's why we've seen, a reasonably high proportion of side effects. And so I think, there's still fine tuning with this mRNA approach before we have it optimized. But again, it's fundamentally different for the reason that I and Joel have explained. The type of antibodies that emerge from the e-vaccination, mRNA or other protein vaccination.
All right, well, I think that brings us to the end of this event. I wanted to once again thank the panelists for a very rich and serious conversation about the science and the opportunities. And I, for one, learned a lot. I'm sure audience members did. And that'll wrap up this edition of the GeneWorx Discovery Series. And thank you all for your time this afternoon. Thank you all for attending. The session was recorded and will be going out. You'll get a link to all attendees and panelists, of course. And see you next time. Thanks, everybody.